The endoplasmic reticulum is a dynamic pleomorphic organelle occupying 10% of typical cell's interior space. It can be divided into a rough and a smooth subcompartment, both enclosing a lumen with a composition that contrasts significantly from that of the cytosol. Numerous structural characteristics are unique to each subcompartment, allowing the rough ER and the smooth ER to fulfill distinct functional roles. Over the years, the differences between the two subcompartments of the ER has fueled the deterioration of relations, and now the two who were once old friends barely share much more than the basic requirements as components of the ER. Little to their knowledge, the smooth ER and the rough ER engage in a range of common activities and share a variety of proteins, making them a lot more similar than they could ever realise.
What the? Why? What are you doing here?
I think you'll find this is my territory, old friend.
Friends? We were never friends. We're too different. Our structure and function, my structure and function is completely out of your league. Plus, you can't compete with my sexy ribosomal studs.
I think you're quite right about the differences, but incorrect about your supposed superiority. For a start, my tubular structure is a lot more convoluted than yours, and my branch points are a lot more frequent, thus helping me form my sponge-like structure that helps me fulfill my many distinct functions. This architectural exhibit is referred to as random tubular. I can see there's nothing random tubular about you. I'm sure you'll be enthralled to know that in some tissues I can form parallel stacks of cysteinae, which can either be lined up against the nuclear envelope or they can be found elsewhere in the cytoplasm.
Pretty pathetic, Smooth.
I think you'll find I'm not quite finished yet. My cysteinae can form concentric whorls or sinusoidal or curved arrays that have cubic symmetry. These are sometimes referred to as cysteloid ER. Furthermore, due to the high expression of the enzyme hydroxyl methylglutarate CoA reductase, my tubules may form bundles with hexagonal symmetry. My different structures are so varied, it led to research introducing the term OSER, organised smooth ER. This is used to describe the variety of impressive structural organisations I can form.
You can't compete. You're lacking the presence of bound ribosomes, one of the staple constituents of rough ER. Under both light and electron microscopy, this is the defining feature of my structure. Associated with my bind ribosomes are proteins known as translocation channels, or translocons. Translocons are composed of more than 20 polypeptides that span my membrane numerous times, and that's only the beginning. Associated with my translocons are the translocon-associated protein complex, translocating chain-associated membrane protein complex, oligosaccharide transferases, signal peptidase, the signal recognition particle receptor, and accessory proteins. Furthermore, proteins exist with the translocons Sec61, alpha, beta, and gamma proteins, which provide key roles in ribosomal capture. The structure may be tubular, but I have a sheet-like appearance. The low curvature of my membrane results in this sheet-like morphology. Rather than tubules, this is what is thought to determine the ribosome in translocon aggregation in the rough ER. The formation of such sheets isn't well understood, but it has been understood that chaperone proteins from a matrix that serve as a scaffold for membrane proteins. Alternatively, proteins are thought to form a bridge, separating the membranes at a constant distance.
The formation of sheets may be poorly understood, but the formation of SCR tubules is thought to involve the action of the cytoskeleton. That's how important I am. Molecular motors are thought to pull out SCR tubules from membrane reservoirs and transport them along microtubules or actin filaments. The cytoskeleton is therefore central to my elongation.
Yeah, whatever.
The shaping of the tubules is thought to be due to the exclusive presence of DP1 proteins. Overexpression of these results in long unbranched tubules. Oh, and you'll be interested to know that although I was once considered a lesser cousin of yourself, recent research has revealed the presence of subdomains within the SCR, including ER sites, the peripheral ER and the mitochondrial associated membrane. So really, some might argue that you're the lesser cousin.
They're called subdomains for a reason. They're not important. You may think your structure is critical because of cytoskeletal influence, but my ER membrane is a continuation of the nuclear envelope.
Oh, but what you don't know is that my ER membrane is also a continuation of the nuclear envelope. Even the lumen of my peripheral membrane is a continuation.
Oh, don't go thinking we're all that similar. My lumen thickness is 36 nanometers. Yours can't be anywhere near that.
Oh, Well, on the contrary, mine is 38 nanometers. But maybe we're more similar than we first thought.
That may be the case in regards to structure, but I can assure you my functions are completely different to yours. I'm considered the protein powerhouse of the cell, and considered by most to be of far greater significance than your ribosome lacking self. My most well understood and widely known function is the synthesis of secretory and membrane proteins, utilising the ribosomes bound to my membrane. The synthesis of these proteins takes place after a messenger RNA binds to a free ribosome. Polypeptides synthesized on membrane-bound ribosomes contain an N-terminal signal sequence, including a stretch of 6 to 15 hydrophobic amino acids. As the signal emerges from the ribosome, the hydrophobic sequence is recognized by a signal recognition particle, the SRP. This consists of 6 distinct polypeptides and a small RNA molecule. SRP binds to the signal sequence and the ribosome, preventing further protein synthesis. Bound SRP allows the entire complex to bind to my membrane through two interactions, one between SRP and its receptor and the other between the ribosome and the translocon. Translocons allow the nascent polypeptide to pass into my lumen. Once the SRP ribosome nascent chain complex binds to the ER membrane, the SRP is released from its ER receptor and the ribosome becomes attached to the cytosolic side of the translocons. You listening, mate?
Vaguely.
The signal sequence of the polypeptide is inserted into the channel of the translocons, and this displaces a helical plug, allowing the growing polypeptide to be translocated into my lumen. Once termination of translation has been achieved and the passage of the protein into my lumen has been accomplished, the ribosome is released and the helical plug is inserted back into the translocons. Impressive, huh?
So basically, what you're trying to say by using big words and silly names is that you just help make proteins.
I not only assist in synthesizing fresh proteins, I also play a critical role in the processing and modification of polypeptides. Once in my lumen, the signal peptide is removed from the polypeptide by the signal peptidase, and carbohydrates are added by the enzyme oligosaccharide transferase. Unfortunately for you, you're lacking in both of these enzymes. My lumen also contains a variety of chaperone proteins that assist in the folding of unfolded or misfolded polypeptides. An enzyme, protein disulfide isomerase, or PDI, also found in the lumen, is responsible for oxidizing cysteine residues on polypeptides, thus forming a disulfide bond.
Get your hands off me! Unlike you, I may not have a role in the synthesis of proteins, but in the liver my function is paramount. The lumen of the SCR contains enzymes that catalyse a series of reactions to detoxify harmful drugs and metabolic compounds. The most profound of these is cytochrome P450 family of enzymes. These are responsible for the catalyst of a series of reactions in which water-insoluble drugs or metabolites are prevented from building up by being rendered water-soluble. Oi! Don't be so rude and listen to me. This allows ability to leave the body via excretion. When large quantities of certain drugs enter the circulation, certain detoxification enzymes are required in vast numbers. In response to this, I'm able to double my surface area in a matter of days. Once the drug is removed, the excess membrane can be degraded by autophagocytosis. Also, do you know much about calcium ions?
Of course I do, I'm not an idiot, man.
Well, I don't want to make you feel bad, but I play a really important role in sequestering calcium ions from the cytosol. Its release and subsequent reabsorption is controlled by a number of extracellular signals, and it's pivotal in many cellular communication pathways. In skeletal and cardiac muscle, I've even set aside a region of my reticulum for storage of calcium ions. This is known as sarcoplasmic reticulum. Calcium ions are released from the SR via ryanodine receptors and sequestered from the cytosol using calcium ATPase in the sarcoplasmic reticulum, known as SERCA. Furthermore, research has indicated that calcium ions are sequestered in subcompartments of the SR that can essentially function independently. The release and reuptake of calcium triggers the contraction and relaxation of myofibrils during each round of muscle contraction.
Okay, that might bear some importance, but you're not the only one involved in cell signalling. All the proteins synthesized on my membrane-bound ribosomes then become glycoproteins. As I'm sure you're aware, this group of compounds plays an essential role in cell signalling and recognition. The addition of sugars to a protein is a highly specific process and is catalysed by a family of membrane-bound enzymes called glycosyltransferases. These enzymes catalyse the transfer of a specific monosaccharide to the growing added end of a carbohydrate chain. The sequence in which the monosaccharides are added to the chain is dependent upon the localization of the enzymes within my membrane. The core segment of each carbohydrate chain is not added to the polypeptide, to the protein, but is instead assembled on a lipid carrier that is then transferred to the polypeptide. Interestingly, this process is ubiquitous in all mammalian cells. The glycoprotein can undergo specific modification. Defects in this process are known as congenital diseases of glycosylation. An enzyme called GT undertakes quality control. This recognises misfolded glycoproteins by detecting exposed hydrophobic regions. ER chaperones then give the protein another opportunity to fold.
Oh, I also have a role in cell signalling in regards to glycosylation and calcium signalling. I wonder if we have any more similarities. Not to say that we're similar in any way.
Don't jump the gun, mate. Don't go thinking we're best mates again. I've got some pretty specialised proteins in my membrane. There's this one apoptotic one called...
No way, don't tell me. Not BAP31 by any chance, because that's also present in my membrane. This protein enhances a pro-apoptotic Ca2+ signal that passes from the ER to the mitochondria after BAP31 is cleaved by apoptotic initiator caspase 8.
Oh my god, I love that enzyme! Well, I mean, it is one of my favourites.
You're not by any chance involved in lipid and cholesterol synthesis?
Actually, man, yeah, I am. Using acetyl-CoA as a starting point, a series of enzymes converts it to HMG-CoA outside of the ER. This can then be converted to mevalonate by HMG-CoA reductase. This enzyme bound to our membrane. This enzyme is critical in regulating the rate of cholesterol biosynthesis and is controlled by the feedback inhibition, rate of gene expression, rate of enzyme degradation, and phosphorylation, dephosphorylation.
Oh, and I don't suppose the enzyme scarly monooxygenase is also present in your membrane. That's another important enzyme in the synthesis of cholesterol.
Yeah, of course, man.
Oh, wow! We have so much in common, more than I ever realised, old friend. I also synthesise a whole array of membrane lipids. You don't perform that same function by any chance, do you?
Yeah, actually, I do, man. I synthesise sphingolipids, phospholipids, and sterols. The proteins involved in the synthesis of these compounds are integrated into our membrane and have their active sites facing the cytosol. Newly synthesised phospholipids are inserted into the cytosolic side of the plasma membrane. The proteins ORM1 and ORM2 are highly conserved and regulate lipid homeostasis.
Who would have known all this time we were so similar? We focused on our differences and it's driven us apart.
Oh, come on, I'll show you how I hood rat hug.
Oh, you! Well, what you don't know is that my ER membrane is also a continuum of the No, don't tell me. Is it BAP31? Is it BAP31? Fuck. I can't remember what comes after that. The shaping of the tubules is thought to be due to the excessive present... Another important enzyme in the synthesis of cholesterol.
Yeah, of course, man.